thank you.
Is Professor Bian here?
Yeah, I'm here.
Professor, sorry. I'm sorry, I think I introduced you as a lady. I apologize. I was having in my mind your student, I guess. Okay, so can you please tell us a little bit about your poster in few words, like keep it within 2 or 3 minutes.
Okay.
You can share.
I can't share the screen.
You can't. Lawrence, can you share the screen of Professor Bian?
No, Brim has to take out her.
Brim, please stop sharing.
Okay.
Lian. That's Lian. Sorry. All right, Lian Li from China. Oh, very interesting poster. Yes, beautiful. Thank you.
Yeah, can you say
please tell us.
Okay. Good afternoon, everyone. I'm glad to be here to give you a poster about our recent results. Our main results is about to characterize the moisture absorption process of rebaudioside using near-infrared spectroscopy and Aquaphotomics. So, can you see the PPT, the PDF?
Yes, yes.
Okay. You know, RA is a very popular sugar now in China, with its high, because it is has lower heat and toxicity. So it is now used in pharmaceutical science and food industries, but it has its disadvantages. For sometimes it is very easy to absorb the water, which makes it very difficult for formulation application. So in our present study, we would like to try to understand the absorption process caused by the water. So we prepared a sample and first dried at 105 centigrade degree for 6 hours. Then it was poured into a constant temperature and humidity. The samples were divided into 2 parts. One part was used for the NIR spectral acquisition, and the other part was used for the reference analysis. In this study, we used the other part was used for the determination of the water content. Then the spectra we acquired were utilized to analysis with PCA to the cost analysis method. And we could see the results on the right. This figure shows the NIR spectra, and what we focus on is on the combination band. And as we discussed with the committee in the email, this band is normally we use this band because we investigate the solid states, so we can get a spectra with lower intensity. However, when we collect liquid spectra, this spectral range intensity it will be very high, so it is hard to analyze the water species. Therefore, in this study, solid state was used in help us to understand more, help us to get more details about this band. First, PCA was used to analyze the NI spectra. From the spectra, we could find that as the time goes by, the score first increased sharply. It will be constant at about 4 hours later. And the loading, from the loading, we could find a broad peak centered at about 1921. This may be attributed to the surface water. They defined the surface water may interact with the powder, and it may contain different species of water. So we investigate the PC2 and the loading 2 later. From loading 2, we could find 2 peaks. One is 1905, and the other one is 1944. 1944 may be attributed to the bounded water. So during the whole absorption process, we could dedicate that first, surface water play a key role, and it interacted with the powder interface. Then bounded water was bound to the surface water, and at about 4 hours, the water will be saturated. That's the whole process. But what's the details we want to see? We use 2D cost analysis. And this figure shows the results of 2D cost analysis. From the central nodes results, we could find that at first, during the first hour, the auto peak is about 1901. And at the 1 hour later, about 2 hours, it moved to 1936. It means that during this process, hydrogen bond was formed, which moved the wavelengths, which forced the wavelengths moved from 1901 to 1936. About 3 hours later, the auto peak is also at 1936, which means that the absorption process was constant, and the water and the surface water maybe didn't change anymore. And then we analyzed the on-system results. We could find that during the first hour, the change increase order is 1901, then 1895, which means that surface water plays a key role during this process. Then from figure B2, we could find that during this process, 1944 was first changed, then is 1905, which means that during this process, maybe bounded water plays a key role, but surface water also plays an important role during this process. And from figure C2, it doesn't change anymore, which means that after 4 hours later, the absorption process was finished. And from PCA and 2D cost analysis, we could conclude that during this whole process, surface water plays a key role, and then is bounded water. When bounded water, after about 4 hours later, bounded water and the surface water were saturated, the whole process was finished, and which maybe provide a new perspective for us to understand the whole process. And this is our preliminary results, and we will do more result about the peak position in the future. That's all. Thank you.
Thank you so much. I absolutely love your poster, and I want to be the first to comment and say something. So, what you touch about this surface water, and this is the most important thing I think regarding the preservation and water activity and all the interaction with the environment. And it is there is one paper a very long time ago written by Professor Giangiacomo, Italian scientist, who did a paper is called osmotic dehydration in apples. They did a very similar thing, but and from that paper, I first realized that there is a difference between this band that you use, the combination band, and the first overtone of water. So what they noticed is that first there is a change at combination band around I can't remember now correctly, around 1936. And then after that, there is a change at 1455, which we call something like absorption water now in first overtone. And then what we usually call free water, 1410, something like that. The point is what I wanted, I would really like, since you are one of the rare people that use 2D correlation spectroscopy at this conference that you're presenting, I think it would be fantastic if you can also, you showed on your spectra that you used, that you have the whole spectrum, basically. So if you can do the correlation between different regions, between the combination band and first overtone, so we could know exactly which water species is changing first, which second, because you have beautiful time series.
Yeah, very good suggestion. Thank you. I would do more research about the overtone region. However, when we get the spectrum, you find that there will be some very strange peaks I can't understand well. So we use the combination best. Maybe it was interfered by some chemical bond or some other thing. I can't understand this.
I see. A pity.
Yeah,
I hope.
but I will try and find what causes this strange peak here.
Professor Zenko?
Yeah, I think different near-infrared is an overtone, right, region. But within this region, there are small regions that have also their own specificity. So, and you have this beautiful data, so you can spend time on going from region to region to see how different things you can see. For example, 11-1300 is very interesting region, which we think is a second overtone of the one that you're looking at. And it is related to electroconductivity and electrical properties of the media. It is very much useful region for bacteria studies and for sugars as well. So that might be very interesting to visit that region as well. Then when you go to the second overtone, 940, 960, or 740, the third overtone, then you will see electron transition added to this. So each region will enrich your findings. And because you have this beautiful data, and I think we don't know, this is a new sugar, obviously. Nobody has heard about, at least I haven't heard about this.
You were not on diet. This is good for people who have problem with glucose.
Yeah, that was my point. Let's understand why it is good for diet. Because, for example, I think that the obesity is not about fat. Obesity is about water in the body that cannot be released. So, if this sugar is good for the diet, probably this sugar does something to the water that makes it easy leaving the body. So very high potential study, and I am really happy to see two-dimensional and time series. And I would like to, I can see that you have affinity to different analytical methods, multivariate methods. I would love to see in aquaphotomics also doing time series analysis, because from engineering point of view, with the time, you have a huge matrix, three-dimensional matrix. You have wavelengths, you have absorbance, and then you have time. Yeah. And you can do parafac as a simple multivariate analysis, but then you can do time series analysis that will also show you lots of assignment, lots of bands popping up. So then the picture will be more and more clear, I think.
Okay, thank you for your suggestion. Thank you.
Thank you so much.
It's very serious work. Thank you.